So, so welcome to CASP14. I am Christoph Fidelis, Prediction Center, UC Davis. As you may know, we have been doing this every 2 years, recently alternating between Europe and the Americas. And 2 years ago, I had the privilege to serve as local host in Mexico. This time around, I thought we would have one of our European colleagues do the honors in sunny Italy, Spain, or south of France. Well, due to the pandemic, it's my turn again, and I am serving as the local host. I just didn't know how local and confined this will really be. On the other hand, this turned out not to be so very local. As you can see, we have participants pretty much from all over the world. And I think we're cutting out Australia here, but all 6 continents, except for the 7th one, of course, are represented. So quickly to the logistics. We have multiple platforms. If you are hearing me now, that means that you are on the main session platform. So that's already a success. But our hub to kind of link to all the other supporting platforms, we try to make it as much as a real life meeting as possible. We have this web page. You have received the document with links to all the other platforms that we are using. So the conference web page then contains links to the program, conference updates, which is our regular Prediction Center message board, but also a link to this very document which does not appear here just yet, as well as the AirMeet and Discord platforms. And I will talk about this in a second. The sessions that are dedicated to posters will be on Tuesday, Wednesday, and Thursday. And if you click at any of these links, you should be able to see the links to the posters as they are on that web page. So then if you expand this link, you can see the poster abstracts, authors, and affiliations, but also a flat file poster PDF or JPEG file, or a video. In this case, it's usually the poster or a video. In this case, the video is not uploaded, but we see the poster. Then the next platform that we are going to use is primarily dedicated to the live poster sessions that, as I mentioned, will be held on Tuesday, Wednesdays, and Thursday. And the posters are assigned to these AirMeet tables. You have a little bit of guidance about this in this document that was sent. And you essentially enter a breakout room, which can accommodate up to 8 people. And we expect that the presenters of posters will be at their assigned tables. Tables for posters are assigned from number 3 to number 54. And if you have any problems with this, we also have junior scientist volunteers manning desks 1, help desks 1 and 2. Actually, should be there now if you have any questions. But the AirMeet platform is also meant to allow interactions between conference participants. You can see the list of attendees on the right-hand side here. You can use the chat channel, which will remain open throughout the meeting. And you can also use direct chat to send questions to the attendees that you see in the attendee list. You can also enter rooms that have numbers greater than 54 and have meetings on any topic there. I would like to thank you, junior scientists that have agreed to help us. They are from 3 laboratories, one in Gdańsk, Poland, of Adam Liwo and Adam Sieradzan. Also from the group of Maya Topf, this is Tristan, Manaz, and Karen. And we have Sohi and Menjay from the group of Chok Sok. Now, a few words about satellite sessions. So the program is very packed, but we wanted to allow additional topics to be addressed outside of the proper meeting of the main meeting, and for this we have we have suggested a mechanism to allow ad hoc sessions on topics of interest. So these could be set up before or after the main sessions. And some of them are already being set up. We have one on impacts of models on biology on Tuesdays, and data-guided protein structure prediction on Thursday. And to do this, the best way is to go to Discord, and there's a special satellite sessions channel there, and there you could suggest a topic, provide a link to the platform of your choosing, and then suggest date and time, and see what kind of interest you generate, and if you have enough participants to start such a session. We will also try to help posting information about these sessions using other means. So let me just quickly go to Discord and show you this satellite session channel, which is here on the left-hand side. And that's, I think, pretty much it. I would like to think I feel much more confident about doing all this. I would call him brains behind the technology here. It's talking about Seth Mulholland, who is helping us out. He's from ISCB. And I also would like to thank Tom, Jonathan, and Niola from Science and Technology Facilities Council, who are helping with this large Zoom webinar platform that we are using right now. And you see the other 2 providers of help to us to organize this meeting on this page. So with this, I will pass the mic to John. Thank you.
Hello, I'm John Moult. Welcome to this 14th round of CASP. I think everybody on this probably knows a lot about the subject already, but there are a few people who are new. And so I will fill in a little more detail than I usually do in this introduction talk. The other thing I'm going to do that I don't usually do is to give away what the main results are. As we'll see as we get through this, there are some really astonishingly impressive models this time, and that will influence what we talk about throughout the entire meeting. And so I want to set that up as I go through the material here. So we're about trying to advance solutions to the protein folding problem. How do you get from an amino acid sequence to these exquisitely beautiful folded structures? And can you do that computationally? The way we do that is very simple in principle. We take experimental versions of the structures as a gold standard, and we find members of the experimental community who are about to release a structure but haven't done yet. We give the sequences of those proteins to the participants in CASP, who compute their version of the structure, give it back to us, and then there is a comparison of the computed structures with the experimental ones. We're trying to maximize 4 things, really: rigor, and so this is done without the answers being known; transparency, all of the answers are public; collaboration, everybody works on the same thing; and communication, often very intense at the conference, and we'll see what we can do virtually. So there are some requirements to make this work beyond the simple principles. You do need many targets. We've got, I think, 98 this time that are being evaluated in the main section. Each one person taking part won't get considered unless they try a lot of the targets. You need a lot of people doing it. We've got 98 groups this time. I already mentioned the gold standards. There is a very ruthless, in some senses, head-to-head comparison of methods. Someone can see what's happening. But we do that with clear metrics. And most importantly, we do that with other people, not the organizers, trying to make sense of it. And so we have, as you'll see in a minute, each time independent assessors we ask to look at the data and try and decide what's good and what's bad. Let me acknowledge my fellow organizers. First of all, Anna, who many of you know died about 3 years ago and was for many years a mainstay of CASP and is still very sorely missed. Christoph, my co-founder. Andre, many of you will know very well, since he does most of the things at the organizational level, including collecting targets, collecting the computed structures, analyzing them in the established metrics, and working with the assessors to help them make sense. Torsten, who comes to us from a background both as a very successful assessor in CASP, but also a leader in developing online servers that biologists can use to get structures computationally generated. And Maya, who was added to the organizers because we realized that cryo-EM, which is her area of specialty, is becoming a very central thing in the field. This time's assessors, Nick and Lisa doing topology, Andre, Jonah, and Marcus looking at the high accuracy aspects of models, Dan Rigdon on refinement, Alfonso Valencia and Alba on contact prediction, Choksuk for accuracy, Esri Kassa and our Capri colleagues for assemblies, and then finally Sander and Dima looking at function. You can see that these people outnumber the organizers, and so you can be assured that we're not dominating the scene, I think. Right. I think Christoph covered all of this. The important thing from our point of view is to try and make the best use of these facilities to really generate off-stage discussions. That's what CASP meetings are usually so great for. And this time we're really trying to find ways of making it easier for people to interact. If you have any ideas about how to do this better, if some things aren't smooth, let us know. One thing Christoph didn't mention, what we'd really like to see coming out of this, and one of the few advantages of being online, is that we could generate ongoing interest groups who meet periodically after the meeting. For example, there might be a journal club on new deep learning papers and their applications in computational biology, but whatever you like. And so use the Discord channels to set that up if you're interested in something along those lines. I already indicated this, but although modeling single proteins and domains is the sort of core of CASP, that must mean including other areas. And so these are the areas we're going to cover in the meeting. Each of them down to here is an established part of CASP already. The first is refining. Can you take an approximate model and use essentially physics-based methods to get closer to experiment? With the increasing availability of assembly structures experimentally and the improvements in single structure modeling, protein assemblies is now a very central area, and I think will become even more central in CASP as we move forwards. a lot of the recent progress, in fact, in some sense, or at least up until this CASP, all of it has come from trying to predict which amino acids in a sequence are in contact in the folded structures, and more generally what the distances between amino acids are. And so there's a section on that. Models are not much use if you can't tell the user how accurate they are, both generally and in detail. So session on that. And getting function from models. I think that those of us who are structure wonks think structure is the end, but for other biologists, the question is, how well can you use them to derive aspects of structure? And I think we were fortunate in our choice of assessors, both because they're good, but also because what they do this time, they're going to be telling us about how good the models are for small and large molecule docking. And that's going to be highly relevant to applications of modeling in the next little while, I think. 2 extra sessions, one on the relationship between COVID and CASP. That includes a talk from David Baker on the work he's and his group have done on designing new therapeutics and vaccines. A talk from Andrzej Jachimiak, who is a leader in getting experimental structures of COVID proteins, and a discussion of the outcome of the CASP experiment, where we got the CASP community to produce models of some of the hardest to treat cases. Then there is a session on the future of deep learning. Essentially, all of these areas are now using deep learning. And as we'll hear, deep learning has made a huge impression this time on the accuracy of the single protein models. And so we'd like to bring that together and see where things can go from here on in. There'll be 2 parts to that session. The first is an overview talk on where AI is going from Demis Hassabis, who, as many of you will know, is the CEO and founder of DeepMind, and also in his own right, a leader in the field of artificial intelligence. And then a discussion with people who've tried to use these methods in each of these areas, not to talk about what they've done, but what they now see as the way forwards. So that's where we're going. Let's now talk a little bit about what's happening in CASP at the moment. I showed a version of this slide one CASP ago, and the point I wanted to make was an analogy between punctuated evolution in the history of life on Earth and progress in CASP. The theme being that we often go through quiet periods in CASP and then something exciting happens. And each of these white boxes is one of those exciting steps forward. I've talked about some of them in the past, and you can see they sometimes reflect what we now have as sections. Actually, in the last 4 CASPs, we've been on a real roll. There's been no pauses, no punctuations, no equilibrium to punctuate, I would say. And that's because, first of all, of trying to predict which amino acids are in contact, and then moving on from that, and I'll try to show you how that happened, to applying deep learning methods to contacts and distances. The history of the contact area begins with this paper in 1994. You'll notice Alfonso Valencia is one of the authors, and he's this time the contact assessor. And so it'll be interesting to get his perspective on all of this. And here's a very well-used slide of the principle of this. If 2 amino acids are in contact in a folded protein, that will put restraints on what pairing you can have here. For example, if these are close together, you can only have 2 small things. You can't have 2 things with the same charge, but it would be great if you had things with opposite charge, and so on. If you have a set of evolutionary related protein sequences for the thing you're interested in, you can line those up, that's what this is, and you can look for pairs of columns in this alignment where the residues you see are correlated. There is some connection between what I see here and what I see there throughout the column. And you can use very simple sorts of statistics to look for those correlations. People started doing that immediately after the first paper came out, and we started tracking it, I think, in 1996. And for a long time, it didn't work very well. There are different ways of tuning these things, but if you tuned it optimally, you could get something like 20% of your contacts predicted correct at the price of 80% of them being incorrect. And clearly, that's not very useful. And so these were of academic interest and were used a bit, for a long, long time, in fact, up to 2014, so 20 years after the paper, the methods did not work very well. Two things happened. One was more data, so you could get much better alignments, and that certainly matters, but it doesn't get you above the 20%. What gets you above the 20% is realizing that just looking at these correlations is much too simple a statistical model. One of the puzzles is why it took us so long to get there, but because there are in other areas, in statistics and physics, already solutions to that. And once people started applying those solutions, suddenly these methods became seriously useful. So here's actually from CASP11 in 2014, an example of what can be delivered with these methods. This is a model, this is the experimental structure. You can see the topology, the fold, is very similar. Although when you look in detail here and here versus here and here, you can see there are serious differences. These are not perfect models. They have the fold, the topology right. And in 2011, we really only saw two of these. We had to wait until 2006, sorry, CASP11, and we had to wait until CASP12, 2 years later, 2016, to see the accuracy for many of these structures in terms of predicting the contacts getting much better. So here's a picture of that. This is from the assessor, Andre Fischer. And what you see here is ranking of the accuracy, the precision, the fraction of correctly predicted contacts for each group. So this is the best group on the left. You only need to look on the left. Here's where we were stuck at before the methods improved in CASP10. Here's a little bit of a blip. That structure I just showed you was from this period, 2011. And now people really got these methods to work, and you see the precision is going up to nearly 50%. And that did lead to many more nice models in CASP12 in 2016. But actually it didn't do a lot to improve the model accuracy for all of the structures. We have this rather unforgiving way of looking at the overall performance in each CASP, which I'm going to show you in a second. And you'll see there that the difference between CASP11 and CASP12 in terms of the overall accuracy of the models is not so great. So let's go to that. And I'm going to spend a minute or two explaining this because there's a lot of talk about GDTTS and what it means at the moment. So I'll try and be clear about it. So this y-axis is a measure of how accurately the backbone of the protein is modeled, the backbone being the sort of thing I showed you a couple of slides back. Random is somewhere around 20 to 30. If you just superimpose two proteins that are not related, you'll be somewhere here. Getting the topology right, the thing I mentioned just now, is about 50. So if you're above 50, your topology is probably correct. Each model, each CASP on here is represented by a line and not a point. And that's because different targets have different degrees of difficulty to be modeled. An easy target is one where there's a structure which is closely evolutionarily related, already known. And so just by copying, you can get quite a good model. A difficult one is where you don't have that relationship, and that's where things are much more challenging. One point to mention is you can see these lines don't go to 100 here as the targets get easier and easier. They go to somewhere between 80, 85 and 95. These are of course rough fits, so you can't take an exact number from that, but roughly 90 as the midpoint here. And so and for other reasons, it's pretty clear that once you get above 90, although you're not perfectly agreeing with experiment, you are in a region where the remaining differences may be the model or may be various errors and artifacts in the experimental structure. And I'll come back to the significance of 90 in a few minutes. Anyway, we got here because I was mentioning that each line then is one CASP. This is the disastrous CASP1, but then rapid progress, then slower progress. This is CASP11, and now this is CASP12, where the classical contact prediction methods really kicked in, but with minor effect on the accuracy, even for the most difficult targets where we expect it to make the biggest difference. So that was a little bit disappointing. And also, we thought we'd pretty much saturated what those methods could do. So it was unclear whether we'd really see much progress last CASP in 2013. Methods were great, gave us a little bit, but then as we've often seen in CASP before, you get stuck again. So I was afraid we were going to get stuck. But what we saw was this. I'm sorry, let me go forward a little bit to here. Was this. So we've suddenly gone up. This is now our CASP12 line to CASP13. And you can see that rather than plunging below the 50 as you get to difficult targets, so the topology is wrong down here in CASP12, in CASP13, the line asymptotes well above 50. So for most targets, each target is one of these points, the topology was correct, although we're still a long way from this 90. What happened here? What happened, I think, was people were looking at contact plots. So you can say, well, what does contacts look like as a way of looking at proteins? And so you could take along an amino acid sequence axis here, 1, 2, 3, 200, whatever, and here, and you can mark where the contacts are. And this is, you get all sorts of interesting features. These are helices, these are anti-parallel strands, and so on. But you've essentially got a two-dimensional representation of what your protein structure is. People have been using these since actually I was a graduate student in the late 60s. And they're very useful for checking, how am I doing at contact prediction? What happened was that people realized that this is a two-dimensional image of a protein. And it was by then clear that two-dimensional images are really great for some aspects of AI, particularly something called convolutional neural networks. So this is an example actually from somebody who gave a keynote at the last CASP of using these for a sort of Mickey Mouse, well, Mickey Dog application. The idea here is to train, this is the network structure, to train such that you can predict whether an image contains a cat or a dog. And you give the network a lot of pictures of dogs, a lot of pictures of cats, and you try to predict which is which. Now, if you think about, again, a protein contact map, they're all different for each protein. And so it occurred to people, can you train, put contact maps here, put sequence information here, can you train the relationship between the two? And it turned out the answer was yes. And a number of groups, in particular DeepMind, but other groups were not far behind, successfully applied this finally in CASP13 2 years ago, after a considerable period of development within the CASP community. All right, let me move on from that and talk about what happens next. So what happened in CASP14? And here's where I'm departing from tradition. I'm going to give you CASP14 results on this curve. The first thing I want to mention is what happened with servers. One of the concerns that's always raised in CASP is, okay, this is what somebody can do in a lab with rough software, which they might or might not be telling us everything about. That's not much use to me as a biologist. I want something I can send a sequence to and get back a structure. So if we look at, in CASP, we do monitor that. We do monitor how well the server section is doing. And so I'm going to put onto this the CASP14 server performance. That's this orange line. And you can see it's above the humans 2 years ago. So servers, the sort of thing, although they're not actually all out there yet, you can use as a biologist, catch up within 2 years of what's happening in CASP. Okay, so what about the humans in CASP14? I'm going to put on all but one of the human groups. And that's this green line here. And you see there has been, again, this is last CASP, this CASP, substantial progress, not quite as much out here as we saw in the previous CASP, but actually very nice. And now asymptoting at something like 70 rather than 60, but not at 90. So as I mentioned, there's one group missing from this, and that's DeepMind. So now I'm going to put up the same CASP14 performance curve, including DeepMind. And that's that black line up here. Now, if you look at this line, In the middle, it's about 90. At the hard end, it's about 85. At the easy end, it's about 95. This hard end is pulled down by some interesting exceptions. 2 of these are NMR targets, and I think there's a lot to sort out there. We'll be hearing about that later in the meeting. This one is a 50 tumor repeat of the same structure, where the local structure is dominated by interactions. And a caveat on these methods is that when there's a lot of influence from interactions of neighbors, there will be remaining errors. But actually 2/3 of these points, so these are now the 14 points, right, are above 90. And in that sense, this is a real atomic level competitive with experiment solution to the protein folding problem. This is a really extraordinary set of data. I wasn't sure I was going to live long enough to see this, but here it is. Again, with a few caveats, but the caveats are relatively minor. Let me just give you a sense of what this means in practice. Here's 2 quick examples. This is a 532 residue protein, superimposition of the model and the DeepMind model and experiment. The GDTTS is 92. There are differences, of course. If you look here, you can see that the model in magenta has a different conformation of this loop than the experiment. The question is this a deformation from crystal packing, or is this that there's a model error? So this is what I mean by competitive with experiment. And there are other, you could talk about features. If we go beyond the main chain and look at side chains, we can see also an extraordinarily high level of accuracy. Here's another example. This is SARS-2 protein ORF8. Actually, this is a bad model. This has a GDTTS of 80, not 90. And you can see, for those of you who know to look at this stuff, the accuracy of surface side chains here, excuse me, is really, really extraordinary. So both the general fold and the atomic detail, and in fact, I think the atomic detail is key here, are really excellent in many of these models. Now, some people have been very excited to see these results. Some people have kind of said, oh gosh, it's over. There's nothing left to do. I think that is exactly the wrong reaction. I think that showing these methods can work so well for this problem suggests they'll work very well for the other problems we as a community are interested in, which you're fully familiar. But again, extension to complexes, I think, is very exciting at this point. What it'll do to protein design, excuse me, is very exciting. I think there is scope for doing things with conformational change. I think that we'll be able to look at the preferred conformations of disordered proteins with these kind of methods. We'll be able to do mutation interpretation, and they will lead to better docking methods. Of course, all this has to be shown, and these are future areas for CASP and Capri and other experiments of this type to focus on. But I think this really is an enormous and exciting step forwards. And we're going to hear a lot about it, of course, in the meeting. We're going to go on next to a session where experimentalists have already made use of these models to solve and improve their structures. Then we'll hear today from the 2 assessors in this area. And tomorrow, in the first part of the day, we'll hear talks from 3 of the more successful groups. This is really, really amazing stuff. And I hope we're all going to enjoy hearing about it in the meeting. All right, I'm going to stop now, and I think I hand over to Christoph again to introduce the next session. Christoph, you're muted.
Yes, so the next session will be chaired actually by Andrei. So I guess over to him.
Okay, thank you, Christoph. John just had a great teaser and great introduction to what's happening in this CASP. And he already introduced me. I am one of the CASP organizers, and I'm also a person hiding behind this gear mechanism representing a prediction center for CASP. And as you can see, all the data flows through us, and being in the middle of this data avalanche is laborious, and sometimes it's stressful, but it's also rewarding beyond just being paid for what you do. And one of the rewards here is being able to take a sneak peek at the data at the very early stages. And this CASP was not an exception. So somewhere in the middle of June, first targets started to close, and we received several structures of these targets already from the experimentalists. And when we cracked our evaluation system and received first results, and I took a look at them, so I was surprised to see very high numbers showing the accuracy of the models. And John already mentioned in his talk what is GDTTS, that's the measure of accuracy that we use in CASP. And you see here one of the first targets that were evaluated, and the accuracy was 93 point something, you know, but that was a template-based model and target, so it was on the easier side. So we thought, okay, what will happen with harder targets when they come? And here was the first pre-model and target that we evaluated. Score was 87. Second score was again 87. The next score was 90. So across the board, we saw that scores are really high. And somewhere at as the model and experiment continued to unfold, we started getting convinced that that's rather a trend and not an exception. And all these results were coming from the same group 427 or Alphafold2. And at that point, somewhere in the middle of the experiment, we started tracking progress of this group and tracking their scores. And we noticed that scores from this group are consistently high. And we built a graph, something like this at that point, but now we can see this graph with all the data in. And what this graph shows, that's GDTTS accuracy of models from group 427 on all the targets. And as you can see, this plot is really amazing, because what we can see here that approximately 9 out of 10 targets, this group was able to predict to GDTTS of 80, which is really high accuracy that we use this for definition of high accuracy before CASP14. And then even moreover, almost 2/3 of these targets were predicted to GDTTS of 90, which is really, really high number. And as John pointed out, it's almost the accuracy of experimental structure. So I know that there are a lot of experimentalists here listening to this session. So, and you don't speak maybe GDTTS language. So here is the dictionary. So here is the typical RMSD that you can expect at some levels of GDTTS. So, and as you can see, so 90 GDTTS corresponds approximately to 1.4 angstrom RMSD. And 80 GDTTS corresponds approximately to 2.3, 2.4 angstroms RMSD. So it's really amazingly high accuracy predictions. Okay, so what we did, we took these predictions and we decided let's use them to our advantage to maximize the output of the solved structures. And in any CASP, we have some structures that we know people are working on them, but are not there yet. So we decided, okay, so let's try this. We'll give these people models from group 427, and we'll see, you know, if they can solve their structures or improve their structures by using these models. And this session we'll hear from some of these people. We'll have 4 talks from the experimentalists who took these structures from 427 and used them to their advantage. And also we'll have 3 brief communications. And now I want to introduce our first speaker. It's Henning Tidov, and he is a professor at the University of Hamburg, and he had a typical X-ray problem. He had the data from X-ray experiments, but he couldn't solve the phasing problem. And we gave him a model from group 427, and you can see this model superimposed onto the experimental structure in this slide. And we'll hear his story in a second, but introducing our speaker, I'll be given a short excerpt from the email that I received from them. The very next day, they inspected the structure. So that was a reaction from Henning. He wrote to me, the model you sent me from the leading group worked for molecular replacement, and we finally solved the structure by MR-SAD. I am still astonished that the human expert model worked, while none of the server models we tried did not, as they were rather similar. Great job. So with this, I am giving and passing my word to Henning. Henning, please take from here.
Yeah, thank you very much, Andrei. Sorry, I'm just trying to share my slides. Need to start it again, sorry. Okay, there we go. Okay, I hope you can hear me and see the slides. Otherwise, please shout. Good. Yeah, thank you very much, Andrei, for this introduction. And yeah, I try to stick to the 10 minutes. So we are one of the experimentalists that Andrei already introduced that actually made use of the DeepMind model in order to solve our structure. So by the time we submitted the structure or the sequence to CASP, actually we were sort of already struggling for this to this problem for approximately 2 years. We had nice data. We couldn't really solve it by molecular replacement. We had experimental phases. They were not really good enough. We had a model that was sort of 70% complete, but we couldn't finally finish it. So let me just, sorry. So now we should be in presentation mode. Good. I don't bore you with biology because it's not about biology, just brief introduction of what this is about. It's about iron uptake pathways in gram-negative bacteria. So gram-negative bacteria usually take up iron in a chelated form with the siderophores through the outer membrane and then through the inner membrane. So we determined the structure of the outer membrane transporter, and then finally we found another operon in the same protein in the same operon that's involved somehow in the uptake through the inner membrane. So this protein we called FOXB, and this is what it's all about. So let's just go to the final structure. So this is the structure that Andrei already showed before. We knew already that it probably has some heme bound because of the spectroscopic properties, and the crystals are bright red and so on. Obviously, this I didn't reveal when I submitted the sequence. So this is the final structure here, 2 heme molecules bound in the transmembrane region. So this is probably a reductase, which was not known before we actually determined the structure. So we thought, okay, it could be a transporter, could be a reductase, because both is required for the function. Otherwise, these 4 transmembrane helices and these 2 Pepsi domains that are in contact with each other, these we tried also for replacement, also on its own, it didn't work. Hemes both coordinated with the typical histidine coordination. I compared later some of the server models and the best model from DeepMind that actually worked. Actually, most even of the server models have all the histidine coordination more or less correct. So this was, I'm really astonished of how good the models worked in the end. Also, the coordination that I showed you here on top between the Pepsi domains is actually correctly predicted by many of the server and of course also by the DeepMind model. We looked at the structure in the end, that's the DALI results. So there is some similarity, also not too great, to some other reductases in the transmembrane part. And also the Pepsi domains are pretty much conserved. So there are some good models also for the Pepsi domains. Still, we could not solve anything by molecular replacement before. So this is, I think, the part that's most interesting for you. How did we solve the structure? We had native data to sort of 3 angstrom. We had selenomet data to something between 4 and 5 angstroms. We also had iron anomalous data because we knew that there was some heme bound. Still, this was not enough for experimental phasing. Then we tried a whole bunch of molecular replacement with these individual Pepsi domains, with homology models thereof, with anything that we could otherwise use as molecular replacements models. Still nothing worked. At that time, then I decided to submit the structure that was sort of 70% built. So we had the idea for the transmembrane helices. We didn't know much about the pyroplasmic parts. Then I submitted the sequence to CASP14, and honestly, I didn't really expect much. After a couple of months, I remembered that I submitted something there. I just went to the website. I didn't even know how CASP works, honestly. I just downloaded some server models that I could access on my own. And I think, yes, so 33 server models by that time, the day I downloaded it. I tried replacement with all of them. No solution. Although I have to say, out of these 33, I would guess probably 25 or so looked fairly similar of what we were expecting already from our 70% completed model. So they looked reasonable. There were 5 or 6 that yeah, were clearly probably not correct, but the others looked pretty good, but we couldn't determine anything. So I thought, okay, this didn't work, and we went back to make other crystals basically. And then suddenly Andre sent me an email and that he wanted that there was the best group. He didn't reveal who this was, and he sent me the best model. And I thought, okay, give it a try, do a replacement. And there was a clear solution. I'll show you the statistics in a moment. And from then on was basically 1 day of work to get an initial model and then some iterative refinement, and the structure was done. So this is the output from the first quick and dirty molecular replacement that I was basically running 10 minutes after I got the model from Andre. So TF-Z score of 19, so it's a clear solution. Still, our factors are obviously not ideal. So in the end, we had to build quite some model building and combine it with experimental phases. And in the end, we ended up with 3 angstrom and our work are free of 27 and 30%. So let's look at the structure. I think I can, in the interest of time, I should skip this, but of course, all the selenomet sites are correct. The ion sites are correct. The overall density looks fine in the end. Here I just randomly superimposed our final model in gray, with in cyan is the AlphaFold or DeepMind model, and blue, pink, and yellow are 3 randomly selected server models. And you see that even the server models overall look fairly similar to our final structure. However, still none of them worked for replacement. And I think, well, I was thinking a lot of why they didn't work. And honestly, I cannot give you the detailed answer. Maybe you can find out later. But I think it has probably to do with this loop region here, where you can see that the gray, so gray is our final structure, cyan is the AlphaFold model, or DeepMind model, and here the server models that are used from Baker Rosetta server, Raptor-X, and the Shang server, they are slightly different in this region, whereas in all the other regions, even the transmembrane parts, they look reasonably close to what I would expect. That's the picture that Andre already showed before. So that's the superposition of the DeepMind model with our final model. And I look at, in my opinion, is crazy. I would not expect that somebody can predict anything like this. And I don't know how it worked. Overall, actually, our model is missing quite some parts. So there's one loop here in the bottom, which you can see here on the right, which is included in the DeepMind model, where we just don't have any electron density for this. So we could not model anything in this region. Otherwise, I think I could conclude that the DeepMind model clearly helped us to determine the structure after sort of struggling with it for more than 2 years. I still have no idea how this was produced and why it worked better than the others, but it was extremely helpful for us. And yeah, it was just a breakthrough in this research line in our lab. So with this, I would like to thank the people being involved. Basically, everything that I was talking about today was done by a brilliant postdoc in the lab here, Kesha Joost, you can see in the middle. My major contribution was basically just submitting the structure, the sequence to CASP. And then, of course, I would like to thank the DeepMind people for whatever they did, but it was fantastic. Or the Beamline people in Hamburg for lots of beam time, and on the functional characterization, we are collaborating with some people in France. And with this, I think I should stop here, and I'm happy to take any questions.
Thank you so much, Henning. We'll do questions if there are such at the very end of the session.
Okay.
And thanks a lot for staying on time. So we'll now move to the next presenter. And Henning, if okay. All right, now I can share my screen again. So our next presenter would be Piotr Lehman, and he is an associate professor at the University of Texas in Galveston. And he had a research on the borderline between X-ray and cryo-EM. So he had both some data from X-ray, some data from cryo-EM, and he was working on the RNA polymerase. And when I first talked to him about this polymerase, he said, oh, we are working on this, but we don't know how much time this will take us to solve this structure. And then I sent him models from group 427, and that was his reply. Both my student, Alec Fraser in CC, and I are shocked, stunned by the quality of the model. We need to reevaluate what we do here. Alex remarked that he has just learned cryo-EM, and it is already an antiquated field. My conclusion, you do not need a 10 million microscope to get an accurate model. You need to know people who can model your protein for you, or you learn how to do modeling yourself. Seriously, this is mind-boggling. You would not believe how much effort we have put into getting this structure. Years of work, both cryo-EM and crystallography. I mean, this is really shocking. Piotr. So now, Piotr, it's over to you.
One second. So, here. Okay. Can you see my presentation?
We barely hear you, Piotr.
Oops.
Now.
Yes, thank you.
Okay. Sorry about this. Okay, so yes, indeed. So the models were a complete shock to me, and yeah. So let's get on with this. This will be a bit more biology in my talk, because there's it is also a multi-subunit structure, and it's a complex, and I think it will be probably discussed later on in the conference when complexes are discussed. So I will not show you actually the complete structure of this enzyme, because I don't want to steal the thunder that's coming later on, I guess, in the conference. But I would like to introduce a bit more biology here, and I'll try to keep our struggles, description of our struggles, to a minimum. But I thought I knew crystallography before actually starting this project. I learned a lot of, so I was trained with Michael Rossman, and actually I had to learn things here while working on this project. Okay, so so transcription in the cells is performed by DNA-dependent RNA polymerases. So they generally divide into two large classes. This small single subunit RNA right-hand RNA polymerases, such as mitochondria and some bacteriophages, some small bacteriophages, let's put it this way, such as T7 phage example. And there's also multi-subunit RNA polymerases that have this two-barrel structure, and the catalytic site is on one of these barrels, and so these guys actually do majority of transcription in living organisms. And now, so typical RNA polymerase consists of these components. It has this RNA. There's a core, consists of two subunits, beta prime, that are structurally related. There are alpha subunits that are needed for holding this together, but in reality, you'll see our polymerase doesn't have it, and it actually functions perfectly fine. There's a small omega subunit, and so this enzyme is capable of elongating an RNA template, but it cannot initiate. For initiation, there's another factors depending on the organism. So in bacteria, these are sigma factors, and all of this makes a holoenzyme that's capable of initiating synthesis and elongation. Now, so this is called a canonical RNA polymerase. So canonical, most of them are like this, but again, what is most, so we still only scratching the surface of the genomic sequence that's out there. So in terms of, there's plenty of bacteriophages and other viruses that have all sort of fantastical stuff in their genomes. So some of these phages, bacterial viruses, encode things that do not contain alpha, omegas, or sigma subunits. You just can't identify it, and we call them non-canonical. Because they have new mechanisms of assembly of the enzyme and no promote recognition strategies. So now comes this fascinating bacteriophage. It infects B. subtilis. It has a pretty large genome. And what's remarkable about it, it actually contains uracils instead of thymines in the entire genome. So it actually encodes its own nucleotide synthesis machinery for producing this special base, so that CRISPR doesn't cut it, and so on and so forth. So so in the genome there is actually several genes that are related to beta and beta prime subunits, and so they categorized into two classes. And when we analyze the composition of the particle, we see that they're split into two sets, and we can purify the phage actually and extract what's in the phage particle, and we see that this second set, actually, sorry, the first set is associated with the virus particle. So I mean, this proteins are related to each other, and the beta subunit here, for example, split into two different genes, and they're not next to each other in a genome. They're drawn this way, but they're very far away. And so what happens during infection? The phage infects. Actually, there's this multi-subunit protein is inside the capsid. It gets injected into the cell where it gets activated, transcribes DNA, produces actually new polymerase, the second set what we're actually going to be talking about, and then it does majority of the transcription, and then produces virion-associated polymerase that binds to the particle, or actually gets packaged into the particle, and now this thing can go in effect. Now, we know the promoters that these two enzymes actually recognize. So this is the early promoters, and this is the late promoters, and this is the enzyme that we're actually going to be talking today. The thing that does majority of the transcription late, and this is the promoter for this enzyme. So we can purify this enzyme to high quality, and we identify the fifth protein, and this is what this actually protein looks like. If you blast it, there is only one thing. Okay, this is the protein itself, something that's 98% sequence identical, and the same lengths, something that's 6% identical, and basically from a similar phage. And there's something that's only 46%. Coverage is less than 50%, but that's it. So there's nothing else in the entire gene bank that's like this sequence. So this sequence is very special, but it is a subunit in this RNA polymerase. Now, I'm going to actually, I'm going to move forward a little bit. So this enzyme recognizes uracils in the primer. I'm sorry, in the promoter. It recognizes the template strand of DNA compared to all other this multi-subunit enzymes that recognize non-template DNA strand, and it actually functions on single-strand RNA, single-strand template as well. So that's all special things about this guy. So we should be able to solve the structure of it just by crystallography, right? So it's not a very large particle. It's only a little over 2,000 residues. So without this extra sigma factor, and less than 3,000 should be crystallizable. And indeed, we crystallized this guy, no problem, and we crystallized even in the presence of this DNA template, although we actually the DNA doesn't bind to it. So I mean, it's not present in the crystals actually. So it's a 5S complex, but there's no DNA in the crystals. Now, so I obtained plenty of crystals. We've spent a lot of time on analysis of all of this. This is summarized here. So we shot more than 300 crystals, most of which were heavy atom soaks. And for those who knows what it means, how much time we went into this, more than 50 data sets collected, carefully analyzed. I mean, weeks of data collection and months of trying to get the phases for this. So the best data set is several natives. So the diffraction pattern actually looked fine, and no abnormalities, no pathologies. There's no obvious disordered regions. But I have to tell you, in the 4S structure, without the sigma subunit, about 1/3 of the structure is actually disordered, and it's maybe one of the reasons we couldn't solve it. But in interpretable map, eventually, so I put it in quotes because that's what this best map looks like, and it's one of the better regions, is obtained by SAD phasing using mercury derivative, molecular replacement using a partial model, and multi-crystal averaging on top of several data sets. So this was, then we switch to cryo-EM. Cryo-EM wasn't also easy. So the particle itself doesn't go into the hole at all. You need to use a thin carbon. If you use a thin carbon, you have a strong preferred orientation. You can collect tilted stage, but still you are not, I mean, defeating preferred orientation completely. But what is remarkable with the full enzyme with the DNA, it actually went into the holes of the cryo-EM grids much better, and a little preferred orientation. And this is how we collected this data set that now solved to 3.8 angstrom resolution, and using CryoSpark. And this is on the right hand side here is what the density looks like, fairly interpretable and buildable, and so on and so forth.
1 minute.
Now, 1 minute. So about so how we actually did this, and where this actually came, how the CASP model, how the Alphafold2 models actually helped us. So here's this what HH-preds for the 4 proteins that make up the particle look like. So obviously things that are red actually can be even model can produce it, and it's not too bad. But in order for it to work in molecular replacement, to actually, since we have the density from SAD, you basically remove things that don't fit the density, and you end up with about 30%. And it's actually fine to solve by molecular replacement other data sets. But so what to do with this guy that most of it is really strange. And of course we have this GP226 that there's only 2 sequences like this entire database. Now this is what the model for that missing piece, the thing that is not predicted by HH-pred, and that we were struggling to build. We were building it, but it was very difficult. So in yellow is the group 427 model, and red is our final model. And so initially, while we were building it, I mean, a lot of labor here. So this is actually even 3.8 overall. So this is probably worse than 4 angstrom, 4.5 resolution, and it was a struggle. And then we have this model, and it fits into the density like a glove. So what about GP226, right? So this for this guy, and nothing. I mean, you don't know anything. All right. So this is it consists of 2 domains, and here the rigid body fits basically over the Alphafold2 model into the cryo maps. Again, in yellow and green is our final model. As you see, again, fits like a glove. I mean, so some side chains need to be moved a little bit and so on. So that was stunning. That was astonishing. And my last actually slide is about this template, not template strength recognition. So this is the complete 226, the special sigma factor. These are two E. coli factors. As you see, what is similar is this extended structure, dumbbell-shaped structure, similar, this helix kind of similar, and so on. But overall, the fold of both domains are different, and there's nothing like this in the database. None of this actually both of these folds are unique. So my conclusion, so this is the project was brought to the lab by Maria Sokolova, and who was a grad student at the time when she came to the lab, and she got crystals immediately, and we got decent diffraction pattern. And then since then, she actually got promoted to associate professor, to assistant professor, and she actually gave a birth already, and we're still working on the structure. The cryo was done by Alec Fraser, my brilliant graduate student, and Konstantin Severinov, actually, he trained Maria, and Arina is Maria's student now, also extremely brilliant individual. But thank you very much, and of course, the greatest acknowledgement, actually one of the greatest acknowledgements is to group 427, so Alphafold2. Thank you.
Okay, thank you, Piotr, for your interesting presentation. And we are moving forward right now. Our next presenter would be Osnat Herzberg. She is professor at the University of Maryland. And she had a little bit different story. She did solve her structure. So she had structure of this attachment region to the phage tail, but she struggled with one local area in this protein where she could not fit very well model to the density. And then we gave her models of group 427, and that was Osnat's reaction. Unbelievable. They have predicted residues 16 to 75, which is this area in the protein right here. She said also the prediction includes a different assignment of a cis proline than my original assignment. And it turned out that the predicted version is correct because it enables repositioning of a tyrosine residue in the right place. The change together with another adjustment ultimately results in a 2 residue shift of 20 residues. Osnat, so Osnat, over to you now.
Thank you, Andre. Just a moment. Yeah, do you hear and do you see me now? Do you see the
Can't hear you, we cannot see your slides.
You cannot see my slides. What do I do?
Share screen at the very bottom.
Oh yeah, I forgot to share the screen, of course. Share the screen. Share. Can you share this? You see the share screen?
No.
No. Can you see it now?
Yeah, much better. Go ahead.
Okay, thank you. From the start. Do you see now the slideshow?
Yes, yes.
And do you see the arrow?
We see everything. Please go ahead.
Okay, thank you very much. So the protein that I submitted to CASP14 is actually a fragment of a protein, and this fragment is responsible for the assembly of this kind of branch structure that you can see over here. It contains 4 tail spike proteins, so 4 tail spike protein, which are assembled through this fragment, and then the entire complex is attached to the tail of this phage CBA120. And tail spike proteins are, this particular one have glycosidase activity, and we know now from the work of Piotr Lehmann that each tail spike in this case has different specificity towards a different lipopolysaccharide. And therefore, with 4 tail spikes, this broadened the profile of infection of bacteria that this phage is able to accomplish. So there are 4 tail spikes, as I say, TSP1 till TSP4. Each of them function as a trimer. And the sequence that I submitted to CASP14 is this N-terminal region of TSP4, which is responsible for the assembly through 3 domains. This is about 335 residue. There are 3 domains, XD1 through XD3, that recruit the partner TSPs. And there is an anchor domain, AD, which dock the entire complex to the tail, to the base plate. In terms of sequence homology, there is a remote sequence homology to GP10 from T4 phage, and GP10 is involved in the formation, or is one of the protein of the base plate of the phage, of T4 phage. There is no, as far as I can see, a sequence homology of this AD domain. And when I saw the structure of this fragment, I realized that this will make a very nice challenge for de novo structure prediction for you, CASP14 community, and that's why I gave this, I provided this sequence. The structure was determined initially by MAD method. However, the electron density map of domain XD3 was uninterpretable, and we had to use molecular replacement with better diffracting or better crystal form, then reveal the entire polypeptide chain. The structure is refined at 2.6 angstrom resolution to an R factor of 20.6. And this is how the structure look. It's trimeric, like all other region of the tail spike protein. And indeed, XD1, 2, and 3 adapt a fold folds actually that are the same as the GP10 from phage T4. And several groups were able to predict this fold. What people didn't predict, except one group, is the structure of the AD domain. And what you see over here is that 3 chains come together, interdigitate, and they form what's called triple beta helix, very wide helix. And the group that was able to predict at least part of this structure is DeepMind. And here I just strip this green molecule, one of the monomer, and superposition it on DeepMind prediction, one of their prediction. The superpositioning is done on domain XD1 because you can't superposition all the domains. They are not oriented similarly to the crystal structure, and I have a comment about this. But anyway, what you can see here is that at least part of the chain of the beta triple beta helix was predicted by DeepMind, even though they didn't look at the trimer. They only predicted a monomer, and then they also predicted these 3 beta strand sheet that is the end of this AD domain. And our root mean square deviation in alpha carbon coordinate of this region is 1 angstrom, which is quite impressive, I would say. But look also at the prediction of the other 3 domain XD1 through XD3, in particularly XD1 and XD2 are predicted with spectacular root mean square deviation in alpha carbon coordinate of about 0.4 angstrom. No other group has done it as well. In terms of the trimer association, in the crystal structure, all the domain utilize one single threefold axis, symmetry axis. And this include XD2 domain, which the 3 domains do not interact with one another. And this is impossibly functioning in terms of the complex, because those domains that mediate the interaction with partner TSP has to be able to engage trimers. And just to demonstrate it most easily is looking at the structure of GP10, which also Piotr Lehmann determined. And here you can see that each domain that is analogous to the XD to XD3 domains is utilizing the same threefold axis, a different threefold axis, such that there is a surface on XD2 and a surface of XD3 trimers. Each of them is able to engage another trimeric partners. So I don't understand why the crystal structure restrain this domain arrangement, because we have several crystal forms and all have the same single threefold symmetry axis, but in the functional branched complex, it must be that the arrangement is not like that. Now, where group 427 helped me is in domain XD2, where you consider a region where there are 2 tight turns over here. And the way I trace the chain over here is that proline 239 is built with a cis proline, and proline 236 is built as a trans proline, and this phenylalanine occupy the second tight turn. And when I looked at this structure that was predicted by DeepMind, I saw that this was different. Here, proline 236 occupied this corner with a cis peptide, and proline 239 is a trans peptide, form a trans peptide. When I saw this, there is a shift of 2 residues. It was kind of a eureka, because I had a problem that bugged me, also the refinement, which I will allude to it in a minute, or I'll show it in a minute, but I wanted just to highlight the fact that other group also predicted the domain structure correctly. And this is one of the better one from Zhang TBM, I think it's a server, I'm not sure, with RMSD in alpha carbon coordinate of 1 angstrom. But what you see here, that this relatively higher RMSD is due, first of all, to a trans proline where it should be cis proline, which lead to slightly different loop conformation. But more importantly, the adjacent loop takes a completely different conformation over here. There is also different conformation in this loop. And this is the positions that really had was problematic for me, because its position in isoleucine 247 in a place where there was always residual density map that is shown here in green in all the crystal structure form that I was refining. And I knew that there is a tyrosine 2 residue away from 247, but I couldn't find a way how to build a model. This is 8 residue away from the proline, the cis and trans proline I showed you. And I couldn't see how to build the model. And when I saw the model of DeepMind, it positions that tyrosine in the right place, and there is no more residual electron density map over here. And this is the superpositioning of the two structures, the experimental and the DeepMind structure. And you can see the tyrosine here. The differences here is just because those two, the linker at the N and C terminal of the domain are different than the ones in the crystal structure as I showed you before. So I just want to acknowledge my collaborator. Daniel Nelson is responsible for the biology and the molecular biology of this project. And in my group, I want to highlight in particularly Qinlin Zhao that determined the structure of the selenomethionine protein. Thank you.
Okay, thank you so much, Osnat. We are going to the next presenter. If you can unshare your screen, great. So our next presenter would be Andrei Lupas. And Andrei and his group, they have been supporters of CASP for a long time. So we've been having structures from them for several CASP in a row right now. And this time, Andrei and his group were in a more prominent role in CASP. They were, besides being the target provider, they were also the assessors in this CASP. And when we already knew that we are having great results at hand, we had a meeting between organizers and the assessors, and Andrei said, and as you can see, his target is 1100, so it's one of the latest targets that were released in CASP, so the second from the back. And Andrei said, okay, I have some old data from years, and it's dusted somewhere on my shelves. I have NMR model of the same protein that I could solve, but even with that NMR model, I could not solve the X-ray data that they have on this protein. And then he said, okay, let's release this sequence to predictors and see what comes back. And then when we got model from 427 and we gave it to Andrei, that was a message from Andrei. And he wrote, I cannot overstate my excitement at the fact that Marcus solved the structure of this transmembrane receptor by molecular replacement with the models of group 427 and sent around the coordinates today. So essentially with this, I'll word to you, Andrei. Take it from here.
Okay, share screen. And here we go. All right, I assume you can see the screen.
Perfect, yes.
I will take a spotlight. So I would like to tell you about this archaeal transmembrane receptor from Archaeoglobus fulgidus, AF1503, which was target 1100 in CASP, with great thanks to my department, particularly Reinhard Albrecht, Markus Hartmann, the crystallographer of the department, and Murray Coles, the NMR group leader. So this starts for me 30 years ago at the end of my PhD project on the biochemistry of signal transduction in bacterial chemotaxis. I became fascinated with the receptors and the question of how they transduce the information of binding a ligand outside the cell into the cell for activation of the Ka kinase. And I got the notion that the protein was substantially formed of coiled coil segments. And I produced a prediction program for coiled coils, and I modeled a number of the regions that came out of that analysis. I ran out of building blocks for the model, so I only modeled part of the receptors. And based on my models, I came up with the notion that the signal through the membrane was transduced by axial rotation of the helices in the membrane, in the plane of the membrane. And as I was getting ready to write this up, Dan Koshland, a famous biochemist and editor of Science, came up with a competing model, the piston model, which would work by moving perpendicularly through the membrane. And my rotation model simply became another roadkill. And here, 10 years after proposing the piston model, Dan Koshland reviewed all the possible models. At that time, I was already working at SmithKline Beecham, analyzing genomes for potential targets. And I came across this receptor from Archaeoglobus fulgidus, which was the smallest transmembrane receptor of this type which I had ever seen. Between two transmembrane segments, it had a pass domain as an input domain. And in the cytosol, it only had a HAMP domain. It had no output domain. The HAMP domains are mediators of the signals, modulators, not the actual output. So I still don't know how the signal is read by Archaeoglobus from the HAMP domain, but as you will see, we can do all sorts of neat things with this protein. The first thing we did was use it as a model system to solve the structure of the HAMP domain, which you see here. As I had anticipated, it's a coiled coil. The model I had built is shown in red, the structure in green. They fit quite well together. But there were two important differences between the model and the actual structure. The first is that I got the handedness of the connector wrong. The connector actually went the other way. But more importantly, the protein is not packed like a canonical coiled coil. In a coiled coil, the core residues point past each other in the core. Here, they point straight towards each other. So if we look at this, the packing we had solved was this geometry, but the packing of a canonical coiled coil is this. And you can convert these two forms to each other by axial rotation of helices by 26 degrees. And so I revived my old model from my PhD days and proposed that this was the on state of the receptor and this was the off state. And we did a fair number of structural studies showing that both states existed in HAMP domains. But it was still not clear what the actual global conformational change was because we were solving structures individually, not the whole receptor. And there are many reasons, I won't go into them, to think that individual domains will behave differently than the receptor as a whole. And so I decided to build functional chimeras, receptor chimeras with AF1503 as a transmembrane model protein and intracellular effector domains. This is not the first time this would have been tried. Actually, at the time when we started, chimeras had been built for 20 years. And the reason they seem to have worked is because all these receptors are built along a coiled coil backbone. And so as long as you obey the register of the coiled coils, your domains will fit to each other. And so in principle, you can have a range of different input domains of modulators of the signals, so-called transducers, and of signal generators of output domains like histone kinases, adenylate cyclases, chemoreceptors. And you can, in principle, replace the native input domain with other input domains, and you will get a functional chimera. You can also replace transducers. And so we took our AF1503, which has a pass input domain and a HAMP transducer, and we started plugging different output domains to it. And in fact, we made chimeras with a histone kinase, an adenylate cyclase, and 2 chemoreceptors, all of which were functional. The problem was we couldn't solve the structure of this, and so we couldn't get at a system in which we could relate directly catalytic changes with structural changes in order to see what happened when the signal went through the protein. And so we were sitting on this data set, as Andre told you, a data set at 3.5 angstroms from 2010 onward, and we tried a variety of approaches, but we couldn't solve the phases. And we tried all the standard methods, including solving the individual domains by NMR and trying molecular replacement. None of that worked. Of course, we also do protein bioinformatics and modeling. We built, or I built, a number of models, but nothing looked promising, and we were just not making any progress. And so at a meeting of the assessors, I proposed to file this protein AF1503 as a target in CASP14 in the hope that there would be a computational model which would allow us to solve the structure. And this is the model we received from Alphafold. It's shown in red, and the structure we solved by molecular replacement. Marcus, I believe, tells me within half an hour of trying, is shown in gray. And like Osnat remarked, the amazing thing is Alphafold modeled a single chain. They modeled a monomer within the dimer, and yet they seem to have gotten virtually all the interactions right. That was extremely impressive. And to compare this to other models that were filed, I will show you 2 other models. One really surprised me. You see, all these models that are underlined in ochre are actually the same coordinate set. All the ones in the darker colored are the identical coordinate set. Superimposition in Swiss PDB viewer of 0.00 angstroms. And I don't know how this happens, and maybe Thorsten can have an idea about that. But basically, there's one server in here, Tfold, and they basically all have the same quality. And their model, you see here, is actually pretty darn good. Now, Marcus tells me that none of the models he tried from the CASP14, among the CASP14 models, could have solved the structure by molecular replacement. And you can see that the numbers are really much lower than the numbers for Alphafold, but it's still an impressive model. A few CASPs ago, I would have said, this is amazing, these people nailed it. Right. Now, you see that some groups have somewhat lighter colors. Here, for example, from the Kihara lab group. And what they did seems to have been slight modifications to the basic model. Most of the coordinates are the same, but here the Kihara lab modeled the angle of the helix slightly differently. Unfortunately, it somewhat decreased the values, the GDTTS. Now, the last one that I want to show you is from the Baker group, which was, I would say, the 3rd best model. And I didn't find really any identical ones to this one. you see it's still in the pass domain. It is absolutely excellent. But in the helical domain, the angles are not quite correct. Now, one thing I omitted to mention is that there's a pi turn right here, which bends the helix, and there is a pi turn right here, which bends the helix. And Alphafold got them exactly right. And no one else got these pi turns. Right, and with this, I can thank you to Alphafold for helping us solve the structure, and I hand it back to you, Andre.
Thank you, Andre. Very entertaining. So we have one question that I want to be answered right now. So that's the question to Henning Tido. And it's a question from Arne Allison. Arne asks, what extra did the experimental model contribute over the DeepMind model? Do you have an answer, Henning?
Yeah, of course. Well, for the function, so it's a reductase. So of course, the Alphafold model did not give us any heme groups, for example, and also the zinc atoms that are sitting on top of the heme groups that are most likely, as we guess now, involved in electron transport. So just by looking at the Alphafold structure, I would say the structure is virtually identical. I just calculated the RMSD. I think it's below 1 angstrom. So it's crazy good. But of course, we would not have got the heme group that are important for the function from the Alphafold model. So this, I guess, would probably be the next step to predict something like this. So the orientation of the histidine residues that basically trap the heme groups are spot on, but not only by Alphafold, but also by some of the server models that I showed. Most of them actually got the histidine residues that coordinate the heme groups fairly good. But of course, for the function of the protein as a reductase, we need to see the heme groups, which we knew that it probably has some heme groups from the spectroscopic properties before, but from the sequence, yeah, it's probably difficult to predict, I guess.
Okay, thank you so much, Henning. Clear. So we have many more people who wanted to present at this session, but unfortunately, we don't have much time to dedicate to that. So we'll have now 3 really short presentations about what they learned from the models. And first one presentation would be from Stephen Rees. He is a postdoctoral fellow at UCSD, and also G427 models helped him to solve his structure. So Steve, please go ahead.
Great, thank you very much. Let me share my screen real quick. Okay, so my project was on target 1089, which is a sialidase enzyme secreted by bacteria in your gut. So this protein, based on the sequence, we got a crystal down to about 2.2 angstroms. We knew from the sequence it should have an anti-parallel beta propeller domain that kind of orients around a catalytic center here. And that's what we were expecting when we went to do initial solutions. There were models already available in the PDB for this target, 9 of them very low homology, by about 16 to 26%. So kind of like what we've heard from our other speakers today, there were some models available to try, but they didn't perform very well. This is just a brief image from Phoenix Phaser showing that we're not getting scores that we would really expect, and basically scores for a good solution are shown up here in the corner for reference. So initial models didn't work very well. I tried using Fire as well and some other programs that use established models to try to get a better solution. Scores did improve, but the model did not refine well in subsequent refinement steps. When I tried using an ensemble model where we took 5 models that had similar homology and C alpha trace placement, we were able to improve the scores over what Fire was able to predict. Again, refinement wasn't stellar, but a big reason for this was that the ensemble model was missing large parts of the model, probably about 60% of it. So when we built those in just through auto and manual refinement, the scores did get better. And we were able to solve this protein using this ensemble model, but it never really looked quite as good as it could have at its resolution. So in the middle of trying to get this ensemble to work, I was working with, or Andre reached out to me and said, hey, do you want to submit your sequence for this? I said, sure, we're still interested. Within about 10 or 15 minutes, which seems to be a common story at this conference, we were able to get much better fitting scores with the models that Andre provided from group 427. So scores by and large much better on the initial placement. A single round of refinement after this got excellent initial scores, which we're bringing down now with manual building and refinement. So the 5 models that we received from CASP all worked very well, all very high initial placement scores. Something I was personally interested in was what made this different compared to the other strategies we tried. So shown here in pink is the ensemble model we had initially tried. Again, this was from 5 established PDB models, just where the where high RMSD areas were conserved and kept in the model. Everything else was chopped out. That's shown in pink. Note that the conserved areas for most sialidase enzymes seem to be conserved with what CASP had predicted. What group 427 was able to do was really build out some of these areas which weren't conserved in a lot of the other models, and it did so very well. If we look at that compared to FIRE, which didn't have as good of a fit, we can see that a lot of these areas were attempted to be built, but didn't really fit nearly as well. And there are some still major differences in those conserved areas that we had from the ensemble model. So FIRE did an overall okay job, but the fit was never very stellar. Whereas CASP did an excellent job at solving this data set. So, you know, from a crystallographer perspective, we had a target that did not work well from models that were currently available in the PDB and which other servers couldn't do a nice job of predicting. CASP14 did an excellent job of doing that. And this could be a great tool for any crystallographers that, like we heard from previous speakers, they have this project that's sitting on their hard drives, but they haven't been able to solve them through current methods, or they can't repeat the crystals. We have a few projects like that in-house as well. 2 projects where we've never been able to repeat crystallization, that there's no good models for available. We'd love to get those into the pipeline. We have another one currently now, which has poor phasing currently and low resolution, no models available at all, that we'd love to give a try there. So definitely a lot of potential that we see from experimental applications for this program. And so just to keep it brief, special thanks to those in the lab of Jeffrey Cheng, where I'm a postdoc, for helping with some of the purification steps. The Carson Zengler lab at UCSD as well, who did a lot of the biology for sialidase enzymes. This protein had originally been published last year just functionally with this group. Thanks to Andre at CASP and the group at DeepMind for working on this project and the other ones that we're hoping to push, and the fellowship that was able to fund some of this work. So thanks for the time today.
Great. Thank you so much for such a nice short presentation. So next presenter would be Rhys Grinter, and we have to specially thank him because he is in Australia and it's the middle of the night in Australia, and he is still staying up, you know, wanted to tell us something about the models that he used. So Rhys, please, up to you.
Yeah, thanks very much. I actually had a sleep and I've had some coffee, so I'm pretty well good to go. Can you hear me okay?
Yes, yes.
Great. So thanks, Andre, for the invitation to speak really briefly. And it's turned out to be a really exciting paradigm shifting meeting. So it's amazing to be a part of. So I submitted a little protein that I called BON-A, which is target T1054. And I solved this protein by experimental phasing using selenomet to a reasonably high resolution. But I kind of wanted to see, for a few biological reasons, what modeling might do. And so the modeling was very good. Initially, I looked at the server models, and some of those predicted the structure quite well with a root mean square deviation across these atoms of about 0.8 angstroms. And then I got sent to the Alphafold model, which was pretty spectacular with a root mean square deviation of 0.425 angstroms across these residues here. So the whole protein is about 235 amino acids in length, and it's got this unstructured C-terminus, which was unstructured in my crystal structure, but there were some differences, which I'll show you now. So this is just a comparison of the Alphafold prediction, which is great. It's got 2 of these alpha-beta bond domains compared to the crystal structure. And so you might notice here that the first helix of the protein in my crystal structure was actually disordered. If I go back a bit, and I've just modeled it in, but in the Alphafold, it was predicted to be a stable folded helix, which is the canonical bond domain fold, sorry. And so this is kind of interesting, and it's interesting from a biological perspective. If I do a little morph model between the two, you can see that this unstructured region in my crystal structure is folding into this alpha helix to form the canonical bond domain. So I guess if you were less confident about what modeling can do, you might just suggest that the model got it almost right but partially wrong, but there's a slightly more to the story. So biological characterization of Bon A and structural characterization shows that it actually forms in a ligama in the bacterial cell. It's tethered to the outer membrane of gram-negative bacteria, and it helps in a somewhat defined role in stabilizing and synthesizing the outer membrane. And so in my crystal structure, I had it existing as a dimer, which is predicted to be physiological, and we've got some biological evidence that supports this. And so you can see this helix here of the opposing molecule of the dimer actually stands in for that alpha helix that that the Alphafold model predicted was folded in some of the other models too, and forms some nice contacts. And in the cell, and when you purify the full length protein, the crystal structure was a truncation, so I had to truncate the terminal 20-ish amino acids to get it to crystallize. The full length protein is a decamer, which spontaneously dissociates into a monomeric species, as you can see here. And this is a model of my decamer based on some sex analysis that I did. So I think what's happening based on this, just to cut a long story short, and if you want to learn more about the biology of this protein and a bit more about my model, you can read my bioarchive paper, which will soon be a peer-reviewed paper. I think this might explain what's happening in terms of that shift. This is paused. I'll try again. Is it going to work? I can do it manually. Okay, so I think this might explain what's happening when you have this transition that I see from a decamer to a monomer, where the helix of the opposing molecule in the dimer is standing in for the canonical bond domain helix that's predicted by the modeling, and allowing the structure to dimerize and then decamerize. But when you form a monomeric species, I believe that the model prediction is probably correct, that this helix comes around and forms the canonical bond domain fold and forms a stable monomer in solution, because the face that is shielded by this helix is quite hydrophobic, and I expect wouldn't be soluble in solution. So I think this is kind of a cool thing where you got to the point where the model is different than the crystal structure that I saw, but the model probably predicts some biology which is happening and meshes with a whole bunch of other data I have, and allows us to form more hypotheses and to do further experimental work. So thanks for listening to that. And yeah, it's really cool to be part of this session.
Great, thank you for your presentation, Rhys. Really exciting. And our last speaker would be Valerio Chiarini. He is postdoc at the University of Helsinki currently. So Valerio, please.
Hello, hi, thank you for having me. So I know, do you see my screen?
Yes, we do, go ahead.
Okay, so I'll try to be as quick as possible. I know my time is limited, so I will not indulge myself any further. So let's start. I've submitted a fairly small domain, which is called an intane, as you can see here with a horseshoe fold, which is fairly recognizable and already known. So what does this protein do? Essentially, is able to catalyze the self-excision from the two flanking domains here outlined in green. And this function, this catalytic function, is performed essentially by a series of histidine. The most important of all is histidine 69. We solved the structure by heavy atom SAD, so we did not actually use the model to firstly resolve our structures. But we do have two different versions of the same domain, the first of which is the one that you can clearly see here, and the second is characterized by a few different features. Let's see, okay. So which is basically the flipping of three main histidines, okay? Exceptionally high is the flip of histidine 69. Let's go and see how was the model that, of course, is the one of DeepMind. As anyone else has outlined, I have to admit that it is astonishingly good and is represented here with the orange model. So the model was compared with both the APO, the inactive form with the histidine far from the catalytic site, and the holo active model, so with the histidine 69 towards the catalytic side. In both models, the quality, in both comparison, the quality of the model pointed out that the RMSD is below 2 angstrom, but the small difference that can be observed that can be clearly addressed to the difference in the catalytic residue. So of course here I'm playing I'm being really picky and playing the devil's advocate, but as you can see in the Apple form, the one that we submitted, so without saying the possible ligands, these 3 residues have a single RMSD up to even 4.5 angstrom. That is actually because our structure is the first one deposited, which features the catalytic residue afar from the active site. Because histidine 69 was never reported pointing afar from the active site, therefore the prediction algorithm, which was mostly presumably trained based on the PDB data, cannot predict such inactive conformation. Despite that, it has to be said that in imposing the catalytic conformant shared by all the homologous deposited structure, the prediction managed to correctly orient both side chains of histidine 125 and histidine 48 side chains, which are brought together in the active state by a zinc ion, whose coordination is responsible for the catalytic flip of histidine 69, whose RMSD drops from 4.5 angstrom to 0.7. According to the model. So again, I'm being extremely picky, you forgive me. So while the prediction does not fully reflect the alpha state of the sequence provided, it successfully models an experimentally proven holo conformation, supporting the evidence that the binding of zinc is in fact structurally and functionally coupled to the catalytic orientation of histidine 69. So again, thanks to DeepMind, we were able to not only learn that another biologically relevant conformation could be perfectly modeled, but also it was very useful to have further confirmation of what we already established with crystallography and other biochemical techniques. I hope not to have consumed too much time, and back to you, Andre.
Thank you very much, Valerio. So, and thanks to all the speakers. It was a great session, in my opinion. We, even though we ran over time quite significantly, so there is a question to Andrei Lupas in the question and answer session. So I would encourage Andrei to reply that online if he can. We are taking a shorter break right now. We are taking 10-minute break. So let's meet 2 minutes after the hour for our next session. So thanks again for all the presenters, and see you in 10 minutes.
All right, thanks everyone.